party. I can't come to the dance party tonight, but I'm just still very, very happy to be here and meet all of you and all of your families as well. And I'm here to talk a little bit today about Simons Searchlight. So I think you've probably heard at this point about 3 studies. Heard about Rare X before. You're going to hear about me and you're going to hear about Tiger. And I just want to also note, kind of as a part of this, is that we really want to find ways to really collaborate and also combine brain, I should mention. Really want to make sure that we're finding ways to be really collaborative between all these studies and not feeling like you're having to split your time or make decisions about what you should do or shouldn't do. So if you have questions about that, I'm happy to answer some of those. And also I think that's good conversations to be having with anybody if you feel like you're doing one things but not other things, and how you should be, participating in these different studies, because all of us are asking a lot of time and effort and logging onto the internet and potentially having phone calls and finding paperwork. We recognize that, but hope that it does still provide a good amount of value that the panel, I think, did a really good job discussing last night. So overall, Simons Searchlight, you may have actually heard about some precursors to Simons Searchlight. So we were previously known as Simons VIP, and actually were born out of something called the Simons Simplex Collection, or SSC, that actually was mentioned a little bit last night. So we've been around for a while. And it is an international research study focusing on anyone who has a pretty wide range of different genetic conditions that lead to neurodevelopmental outcomes, including autism, but not specific to autism. Sometimes the name of our funder makes that a little bit confusing, but if your child does not happen to have an autism diagnosis, you're certainly still eligible to participate in Simons Searchlight. All they need to have is a genetic variant in DYRK1A that is either pathogenic, likely pathogenic, although we also do accept if anybody in the room has a variant that is of unknown significance at this point, you're also eligible to participate. So we do have a pretty big team of people. I won't go through every one, but there was one individual who mentioned, who I think is worth mentioning again. She was on one of the last slides that Sarah Glass presented for us, which is Wendy Chung, who's very much our lead PI. So I lead the team who's focused at Geisinger, which leads our recruitment and retention efforts. So when you're actually enrolling in the study, you're usually conversing with individuals who are at my team at Geisinger. But in terms of a lot of the efforts that are going on in terms of what genes we're studying, those sorts of things. That's Wendy Chung, who's currently at Columbia University, but as of July 1 is going to be part of Boston Children's Hospital. So you might notice some transitions, but the Geisinger team is going to be staying where we are. And then we also have individuals who are actually at the Simons Foundation, which is our funder, who lead the scientific efforts of the Simons Foundation quite broadly, or at least the neurodevelopmental disabilities arm of the Simons Foundation quite broadly, who have a lot of input into the things that we do. We also have multiple genetic counselors on our study, largely from both Wendy's team and my team, to help facilitate the study as well. We also have a bioinformatics team and just an outreach and communication team. So overall, we understand it kind of takes a village to help support rare disease communities. So overall, if we think about what the overarching goals of Simons Searchlight also, one is that when we want to understand the sort of detailed medical and behavioral history over time of individuals who have not only DYRK1A, but a range of different developmental conditions. And we do consider ourselves to be very much a natural history study in that we're longitudinal. So we're looking at individuals at multiple time points. So once you are in our study, you would expect that we would contact you at least once a year to get some updated information, or anytime we would add a new survey to our study, we might ask you for additional information. Once we have information from you, our goal is to present that information back to you in a way that is helpful to you. And I'll give you some concrete examples of what that looks like now. That has been something that we've developed more over the last several years than we were able to do in the previous years. As was mentioned before, we also do freely share any data or biospecimen samples to any qualified researchers. And I think Sarah mentioned that historically, that has been researchers who are affiliated with a university and have gone through the whole process of getting an IRB or other things that are necessary to make sure that they have the qualifications to access that data. We are expanding that, although the timeline for that is not necessarily clear, but we're hoping that we can ultimately also provide that data to sort of more biotech startups or pharma companies or companies that may not have as clear of an IRB. But currently, if you know a researcher who's interested in DYRK1A or is working on DYRK1A, that information is freely available to them. We also want to make sure that any researchers who are interested in DYRK1A can be connected to our community and connected to you. And I can give you, I'm going to have a couple slides later about how we try to make sure that our researchers who are interested in DYRK1A can communicate with you and potentially participate in more research. And ultimately, sort of the overarching goal is to promote the better understanding of these genetic changes for ultimately participating in things like clinical trials, drug development, different therapeutics for our kids. So I do want to briefly touch on the funding. So I did mention that sometimes the fact that the word autism is in our title can be a little bit confusing. So our broad funder, our umbrella funder is the Simons Foundation. Now they have an arm called SAFARI, the Simons Foundation Autism Research Initiative. SAFARI is the largest private funder of autism research in the United States. But the Simons Searchlight is not really an autism study. So although autism is in the name of our funder, we consider ourselves to be a genes first study. So if you have a genetic change that sometimes causes autism, even if your child does not have autism, you're eligible to participate. It doesn't matter if you actually have that diagnosis. There are some sister studies that the Simons Foundation also funds. The one that you may have heard of, and I know we probably have some people in this room who might participate in both, is SPARK. So the goal of SPARK is better understanding genetics of autism. So you have to have an autism diagnosis to enroll in the SPARK study. And as part of the SPARK study, they do genetic testing, including whole exome sequencing. And if they were to find an individual with DYRK1A or another genetic change that we study, if that child did not already have genetic testing, they would also then tell them about Simons Searchlight and let them know that they're eligible to participate. We also have Autism BrainNet, which is postmortem brain tissue donation for any individuals who might unfortunately be deceased. And then SAFARI also does provide funding for outside researchers. So if you have a researcher interested in any genetic condition that we study or autism broadly, we do put out funding for those types of initiatives, as well as providing research resources to participants, including the biospecimens that we collect here as part of Simons Searchlight, different animal models as well. And the data that we collect, the data that you very generously spend your time giving us. So just to talk very briefly about Autism BrainNet. So we work very closely with them, and it's for individuals, any people who are eligible are in the United States or Canada or the UK. And these are individuals who have unfortunately passed away and then are donating their brain for science. Any, again, approved researchers, so they're under an IRB protocol, can apply to access the data that they may have contributed. As if they were a Simons Searchlight participant, they can contribute that data and also the tissue. There's no cost to families. It is something that if you are interested in, you can sign up for, or individuals may reach out to you about. So looking at different genetic conditions that we study, right now we have about 175 either single genes, so similar to DYRK1A, we're looking at changes that occur within one single gene that we know cause neurodevelopmental disabilities and or autism. We also have a number of what's called copy number variants. So these might be individuals who have more chunks of DNA that are missing or more chunks of DNA that are added. It may not be isolated to just one gene, but we know that those genetic changes also lead to neurodevelopmental disabilities. So overall, because we have so many genes that we're working on, these are some of the things that we're really trying to focus on. So I think the one that I think about the most is thinking about what's feasible and what's scalable, right? So we want to try to reach as many individuals as possible. And one of the reasons that we think about ourselves as being an international research study is because international is very important in rare genetic research, right? So right now there's a relatively small community of DYRK1A, but if we can make sure that we're connected not only with the individuals with DYRK1A who are in the US, not only the individuals with DYRK1A who can speak English, that can help us expand our numbers. So we are offering international participation. We started out with English only, but we now we do have additional language. So you can also participate in French, you can participate in Dutch, and we are continuing to work on additional languages that are in our queue. So Italian, German, Portuguese have been high at the list. We get a lot of that information from you about what languages seem to be the most important, because a lot of times the family group leaders, so the patient advocacy groups, are the ones that tell us we have a huge population of individuals who speak Italian who are in Italy, or a group of individuals who speak Portuguese. That's how we've identified the order in which to add some of those languages, and we try to expand that as much as we can. Now, sometimes individuals with different languages are only able to participate in certain parts of the protocol because we don't always have valid measures for everything in all different languages, but that is one of our goals. So we really want to make the largest group that we can and make it as easy to participate as we can in languages that families feel comfortable participating in, and try to make those numbers as big as possible. That does go into the standardized, validated, and sensitive surveys. I'll talk a little bit more about this a little bit later, but we do try to select surveys that we know that not only clinicians use, but that researchers use and that the FDA acknowledges. And I think that is one of the pieces of feedback we get a lot from our participants, is that measures like the Vineland Adaptive Survey can be really, really onerous to do. However, the FDA looks at things like the Vineland Adaptive Survey for their natural history studies. So while it can be really difficult to get through and feel really onerous, we have selected it for a reason, and we try to take feedback as well from families about what else are you guys seeing, what else do we need to know to make sure that we're giving you the right surveys at the right times. So in terms of information for families, so this is something that might look relatively new. So if you are a Simons Searchlight participant, you'll notice that you're getting personalized reports on your dashboard, or when you log into your account, you'll get these visual measures that are showing you a little bit about what do we know about your genetic condition, and beyond that, what do we know about your child relative to other individuals who happen to be within the DYRK1A community. Right now, this kind of information is not live updated, so we do it on a quarterly basis. So every 3 months, we do an update of your information and of the information that we know about DYRK1A, and it's available to you as part of your research participation online. In our queue, however, and our awesome our development team, our informatics team, I should say, is working on live updates, so that anytime a DYRK1A family joins, it will update all of your dashboard, and we could immediately give that information back to you. So that's not something that we have currently. Right now it's quarterly, but it's in our queue. So if you look overall, so if you fill out different forms, so you might fill out something called the social communication questionnaire, the social responsiveness scale, the child behavior checklist, or the Vineland. This is, I think I have a pointer. So this might be what you are looking at. So it will tell you kind of what is the average, and maybe what does your child look like relative to the average of other children who also are impacted by DYRK1A. And then we're also, and you'll see some of this data in my presentation later, but also trying to provide you with information about your community as a whole. So again, these are sent out quarterly. And so we'll say, okay, here are the ages of the kids that are in our cohort right now. What kinds of developmental and behavioral concerns are they seeing? What types of neurological problems, gastrointestinal problems that you see within the DYRK1A condition. A lot of the data I was kind of compare as Sarah was presenting too. It looks like we're seeing a lot of the same things, which is good. That the information that we're getting from families is the tracking across different studies. We also have blood donation, and I'll tell you a little bit about how we use that blood and how we share that blood. But I do want to just emphasize right now too that we do have blood samples available, blood collection available today. We've actually partnered with Combined Brain. So if you were interested in both of those studies but you're worried about your child being able to tolerate phlebotomy, you don't want to have to stick your child more than once because none of us want to do that, you actually would be donating for 2 studies today with just 1 blood draw. So it's both combined brain and Simons Searchlight. You can do both of those studies today if you wanted to do biospecimens. Both of those tables right out there. We've got 2 wonderful phlebotomists and a team of people who are participating today. So if doing the biospecimen piece is something that you'd like to do, but you've been a little concerned about it or not quite sure, this might be a way to kind of get a little bit more bang for your buck too, and then not make your child go through 2 blood draws or 3 blood draws, but just donate 1 time and actually contribute to both combined brain and Simons Searchlight. If you want to participate from home, however, that's also an option. So when you sign up for Simons Searchlight, we talk to you a little bit about how you can donate a blood sample. We mail kits to your home. We partner with Quest Labs. So that's a national laboratory. We'll help you identify a Quest lab that's close to your home. You'll call them and make an appointment, and then the lab will take care of collecting the blood and then sending it back to us. So how are they used? So this is, I think, a really important piece. So any biospecimens that you donate are freely available, again, just like the data that you contribute to any researcher who might be interested. For those of you who were at the scientific roundtable yesterday, we heard a lot of talks about individuals who are using what's called iPSCs, or induced pluripotent stem cells. So there is a partnership right now between the Simons Foundation and the New York Stem Cell Foundation to create iPSCs. So, and I'm going to jump forward really quickly and talk about what iPSCs are for those of you who may not have been at the scientific roundtable yesterday. But iPSCs allow us to take your blood or your child's blood or their siblings' blood and create stem cells. The benefit to stem cells is that then we can you can turn those stem cells into any type of cell. So for those of you who were listening to one of the talks yesterday, I think it was the very first talk, he talked about creating neurons. So he used blood to create neurons, and that's what allows us to do. We know that DYRK1A impacts the way the brain is developing, and we can use your child's blood then to create iPSCs, which one can create neurons. So then researchers can use that to better understand how the lack of DYRK1A impacts the way that the brain develops and impacts neurons. But we also know from another talk that I heard that DYRK1A affects the GI system. And I think that's feedback that we got at the last family meeting. So they can create stomach cells or intestinal cells to try to better understand that. So iPSCs are a really powerful tool that we're trying to create a biorepository of as part of this. The major limitation to iPSCs right now is that they're pretty expensive to create. The process to take blood and turn them into this type of cell can be expensive for researchers. Oh no, yikes. Okay. I think I'm being told that I should hurry up. Okay. Slides are oh no. Okay. Slides are in a different order than I thought they were. So I'm going to have another slide later that's going to discuss this. But right now we have created some iPSCs. The Simons Foundation, in partnership with the New York Stem Cell Foundation, is actually creating these on the front end for researchers. So researchers can access iPSCs, a number of iPSCs for DYRK1A for a pretty nominal fee. It's just a little over $100 in order to access some of our iPSCs, which is far, far, far less than it would cost researchers to take their blood to the lab and actually create it on their own. So it's a resource that we're hoping will be very, very valuable, especially for some of our lab-based basic researchers who would like to show some data for iPSCs or need preliminary data so they can then go on and get funding for these larger grants. What we know about research is if you don't have preliminary data, it can be really hard to get research grants, especially research grants that are from major funders like the National Institutes of Health. So we're hoping that some of this data, and especially our iPSCs that we're creating that are available at a minimal cost, can help some of these researchers, maybe postdocs or new researchers who need to show preliminary data for future funding, that it's not coming at a significant cost to them to be able to produce some of that data. Okay. Briefly, I'll also talk about Research Match. So if you're part of Simons Searchlight, you also end up being part of Research Match, which goes beyond just releasing your data to other individuals. It also allows other researchers to get in touch with you. So we have a team of people who, if a researcher says, I really want to work with DYRK1A and I'd like to send out my survey to a bunch of individuals, or I would like to invite individuals with DYRK1A into my lab, we'll help facilitate that for you. So if you're part of our research, our Facebook page, we will sometimes post advertisements for other studies, but also will facilitate things through a research match process. It's not, you're not obligated. If you get an email and said you're eligible for another research study, you're not obligated to participate in that. It's just, we think about it as being informational. And if you would like to participate in more research, you have that option through Research Match. Another important piece here, and I think we get sometimes a lot of questions about the number of genes that we're studying, but I think this is what you'll see across, pretty commonly across us, across Tiger, across Combined Brain, across Rare X, is the value and importance of studying multiple research studies. So what we're thinking about is DYRK1A is a fairly small community, at least right now, with what we know. However, we are learning a lot about different genes that cause neurodevelopmental disabilities, and there's been increased focus on this concept of pathways. So there might be, and I think our first researcher at the scientific roundtable yesterday talked a little bit about this, which there are some genes that they operate, while they're not DYRK1A, they might operate in similar ways. And if we can understand how these genes might relate to each other, how they might both impact development, we can actually increase our numbers. So there might be other genes that function in a similar way to DYRK1A, and then we can use the power of adding another gene group to your smaller group to help us get numbers not only for research, but also clinical trials. Okay, so I want to talk briefly about how participation works. So currently we have 66 individuals who have created an account. It means that there's a parent or a guardian or an individual with DYRK1A who signed up online and said, we have a genetic change in DYRK1A. In order to participate in our research study, we do require, though, that everyone submits us their genetic lab report to show that they actually have a genetic change in DYRK1A. That's a required component. We don't want people coming in who maybe just signed up because they're interested in DYRK1A, but don't actually have the genetic change. We have a team of genetic counselors. We actually have 3 genetic counselors on our staff right now, and that's almost exclusively what they do. Is anytime a genetic report comes in, we look at that genetic lab report and we make sure that you have DYRK1A, that you're with the right group. Like, this is your family, this is your home, this is where you should be. We do accept individuals with both pathogenic variants and variants of uncertain significance, just as a note. So if you did get a lab report back and it was uncertain, we do still accept you into the study. Part of the reason for that is that we also review your variant every single year. So if you did get a variant of uncertain significance, or it was likely pathogenic but not pathogenic, so likely the cause but not definitely the cause, our genetic counselors review every single variant every single year to see if anybody has changed. So if your variant is what we call upgraded, meaning it went from a variant of uncertain significance to pathogenic, we will let you know that information. That happens more commonly than being downgraded. So being downgraded means maybe it went from uncertain significance to benign, or we don't think it's the cause. That happens much more rarely, but we do let you know that if your variant status has changed, we change it in our research database. And we also change it in a platform what's called ClinVar. So I think somebody mentioned earlier, I signed up for all the data sharing, but I don't know where my data actually went. So one of the places your data probably went was a database called ClinVar. So ClinVar is something that is updated by clinical testing labs. So when they do whole exome sequencing on someone, they will let this large database know about what variant they found, what the features were, like microcephaly or epilepsy or any range of features, so that when other labs see the same variant or similar variants, they can help interpret them better. So we also do that for you guys. So if your genetic testing lab did not upload your variant information to this broader database called ClinVar, we do that, and then we also update it annually. After you have that done, so that sounds like it might take a long time. However, we do fast track families through. So if your genetic lab comes up and it says that the genetic lab thought it was pathogenic or likely pathogenic, we just go ahead and have you participate. If anything were to happen and we said we didn't agree, we would let you know. It's really this, if you had a variant of uncertain significance, there might be a delay in your ability to then participate in the rest, because we want to make sure that you're spending your time the way that you should be. We then also ask you for your medical history. For those of you who've been part of Simons Searchlight for a long time, you'll know that this was historically done via a phone call. So you had an hour-long phone call with a genetic counselor. That's, I think, a large reason why we see this pretty decent little drop-off, right? So we had 49 people who had a genetic testing report, but then only about 29 people who actually shared their medical history. So to help combat this drop-off, we now have an online version of that. So you, instead of having to schedule and go through a phone call, which we know can be really difficult, you can do this all online now. Although a genetic counselor might contact you if they have questions. So if there's anything that they didn't quite understand, or if you also upload some other information and anything seemed like it wasn't quite right, we do have a method to follow up with you to make sure that we have the accurate information. But most families will no longer have to have a phone call. They'll just go through the online survey for the medical history portion. We then also have a range of other surveys, and you'll see those in my later slides. And then I already talked a little bit about the blood sample. So right now we have 12 blood samples from the DYRK1A community. I think this is the first year that we're offering to do blood draws on site at your family meeting. So I hope that many of you will take it. Sorry? Second time. Okay, second time that we've offered blood draws. So I hope many of you will take advantage of that, and hope we can continue to come back to get plenty of blood.
So that's one with combined brain.
Yes, it's combined brain and Searchlight together. And then we also ask for updates every year. There's a few different surveys that we update every year. We're trying to build longitudinal data about our families with DYRK1A.
Just a quick question on the update. Is there
Yes.
when you view ours-up and okay, there's so many different things on there. Is there like a quick button we should be looking at?
We list them in priority order. So what we think maybe we would like the most is at the top. So if you do them just in the order on the way down, that's sort of how we've kind of thought about, well, what do we think is going to give us the most bang for our buck? And if you can only do one right now, which one would we have you do? So I would go down that order. Yes.
Is that because it's a new version or?
Well, there is a new version, you're right. But also that survey is really, really commonly used by the FDA. So the medical history and the adaptive scales, those are the ones that really build that natural history. What does development typically look like? And I know the difficult thing about it is that one is just a really bear of a survey to get through.
You guys like accept a copy?
I'm sorry?
Like the file length, would you guys accept it? Because I just did one for the ABA. And I know in DYRK1A, I could just mail them the copy of the one that I did, and they're fine. Would Simons accept a copy, or I have to do it again?
We haven't done that where we're able to accept a copy at this point, because we enter all raw data. So if you send us a copy, we would have to have somebody to like go through and enter all the raw data. And right now we don't have the staffing to like go through and enter all the raw data, because we have our own scoring algorithm and the scores and all that. So for right now, at least, we do have to have you do it again.
That was my same question.
Yeah.
I just filled it out for two different ones, the brain registry and
Right.
all right. So
Yes. Well, and I think that's like a broad conversation, I think, for us and combined, like all these groups to be talking about is how do we make sure that we're able to share these data across studies in a way that's going to make sense. I mean, I think for us, like all the raw data is technically available, right? Is technically in our data releases, which happen every so often that we put out a data release, all the raw data and all the summary data is available. But across databases, it can be a little bit difficult to share that. And here's a summary, by the way, of sort of everything we ask for at baseline. So we only ask for your clinical lab report once, although like I mentioned, we will re-review it annually to make sure that you still kind of are fitting within your home. We ask for a medical history both at baseline and an update every year. We do a a developmental update on a yearly basis. We ask for new information about medications. There are a few other things that help make sure that we can identify you across studies. And I think that's really important for you guys in particular, because many of you might be participating in different studies, is that we have this global unique identifier. And then we also have a lot of individuals interested in sleep. Seizures, of course, are important. The Vineland is every year. The child behavior checklist, social responsiveness scale, which is largely related to autism. And then we've recently had this quality of life survey. And then we have this, what we call the brief developmental update, which is your child walking? Is your child talking? So generally very brief, but trying to get an idea about when developmental milestones happen, particularly as children age. These are some new surveys that we're working on. So we are trying to get feedback from families, and we get a lot of feedback. I've mentioned the Vineland multiple times. I know that there's a lot of frustration, especially from families whose children are not walking, who are not talking, or who are at the younger ages, that there are a lot of questions that don't seem applicable. And so we're trying to find surveys that might go for children who are at younger ages, who are not as far along developmentally, so that we can try to collect some really meaningful data. The one that is coming out soon is this ORCA, the Observed Reporter Communication Ability, which is focused on our kids who are not verbal yet, might be using devices or might be primarily using gestures to try to better understand how those children are communicating with us if they're not verbal. We've got a lot of feedback about trying to better understand behavior problems, basic developmental milestones, and then also this developmental inventory. And we're working on a pipeline as well to collect EEG records. As you know, we already do collect medical records from you, but that's typically on you to kind of send them in. With the exception of your lab report, I did forget to mention that if you don't have a copy of your lab report, we will help you get it. Just let us know. There's a consent form that you can sign online. If you remember who the doctor who ordered it or who did it, where, what, all you really need to know is who you were with. Like, what was the doctor's name? What was the researcher's name? Or what medical center ordered the testing? And you can sign a release and we'll do the legwork for you, because that can be a lot to try to get medical records. We also are working on an MRI pipeline as well for some other research projects that hopefully we'll be able to share. Okay, so this is just a preview. So for those of you who are not participants, this would be what we call it your dashboard. So when you log into your Simons Searchlight website, what does it look like? So this would be kind of what steps you need to complete. Like I mentioned, we try to put things in priority order, and some things lead to the other things. So if you're registered, you still have to be consented. We need to have your lab report approved. Again, this is a little bit more outdated, but before it was, we had to schedule an interview with you, and then it was marked when you completed it. So they try to put things in priority order, and you can just click on begin, 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 and go through the different steps. Okay, and so also, I should mention that you do get Amazon gift cards for different survey completions. We base that based on how long we expect the survey to take you. Okay, so let's talk specifically about DYRK1A. So for the rest of the presentation, I'm going to be talking a little bit, just like Sarah talked to us a little bit about earlier, about what we've seen in terms of the presentation of DYRK1A. For the purposes of this, we're only going to be talking about variants that are pathogenic or likely pathogenic. We do have 2 individuals in Simons Searchlight who are registered who have variants of uncertain significance, so we don't know if they're causative or not. But for this purpose, we're only going to be talking about variants that we know are pathogenic. And we see we have a few different kinds of variants. And here's on these little dots right here, you can see the locations of the different variants. For those of you who were at the roundtable yesterday, we talked a lot about the kinase domain. But we have some frame shift or nonsense variants, a couple of missense variants, splice site variants, and then we have some that are deletions, which are not shown on this graph, but they include DYRK1A, and we suspect that DYRK1A is the gene that's deleted that's causing the developmental delays. For what we know about DYRK1A, we expect that it's what we call a loss of function, so meaning that you have less DYRK1A is what's causing the developmental delays. I briefly mentioned biospecimens earlier, so we do have individuals right now. We have 22 samples from all of you, 6 who have induced pluripotent stem cells available. We have cell lines in 1 individual. 11 who have whole blood DNA, and then we have 4 individuals who have donated their saliva. We aren't currently accepting saliva at this point just because the blood is so much more valuable in terms of research, what researchers are able to do. And in terms of the iPS cells, so I think I've heard people mention an N of 1 so many times at this conference, and that most of you have completely unique variants. So if you, whatever your child's or your family member's variant is, it's very likely that that's the only person that we know of who has that same exact variant. Although we expect that all those variants are all causing a loss of function. Everybody has a different exact variant, although they're all leading to the same outcome. So for these variants, these are the ones that we have iPS cells available for. And again, we really want to make sure ultimately that as many different variants are represented as possible. So we want you to be included in the research. When we do data releases, we want to be sure that your individual family member who's impacted by DYRK1A is included. And the best way we can do that is to try to get everyone's variants. Okay, so here, I'm going to try to be, if I can work this, as efficient as possible. Some of this is going to be a bit of a repeat. So just to give you first an idea, so we have, of the individuals we have 26 who've actually gone through the process of their medical history interview. So it's actually so we have, 60 people registered, about 30 people who've submitted their lab report, and about 26 who've actually done the interview. The online version of that is so new that none of these individuals would be online data collection. They would all be people who actually spoke to a genetic counselor. You can see the age range here. That's one of the major limitations of our data set. And I think the major limitation of a lot of the data sets related to rare genetic conditions is that they really err towards the side of younger kids. We don't know as much about some of these older kids. We do have most of our participants, so of the 26, 22 are 18 or younger. We do have some young adults, one older adult, but for the most part, a lot of the data that I'll be presenting on is individuals who are in the relatively young age range. We have a pretty fair split between males and females, close to 50-50. Speaking first about developmental and behavioral conditions, we can see that the vast majority have some degree of developmental delay. I should also note that these are not mutually exclusive categories, so there might be some individuals who are represented in more than one column. So we have almost everyone indicating that they have some degree of developmental delay or intellectual disability. Most individuals also indicating some degree of language delay or speech impairment. About half currently are reporting an autism spectrum disorder in addition to that. And then we also have some other conditions that are sometimes sort of one-offs or two-offs. Some of this might be related to limitations in how we diagnose these conditions. OCD, anxiety, and ADHD sometimes are harder to diagnose in kids who have significant developmental disabilities. So we can often also rely on some of our checklists to try to understand where those behavior issues occur, even if there's no formal diagnosis of some of these.
Can I ask you a quick question?
Absolutely.
You show 88% have an intellectual disability. Does that mean that 12% don't have intellectual disability, or perhaps they're too young for it to show yet?
It could be that they're too young to show. We do also have a learning disability category. It could be that there are some kids who are borderline and maybe didn't get that diagnosis. We do anticipate that almost all individuals are going to have some degree of learning impairment, if not a frank intellectual disability. Additional medical issues that we see. So I heard a lot about this yesterday. So we have about a majority of individuals showing GI issues, about half indicating some kind of allergy, and then less commonly some of the other issues. So respiratory problems, genital abnormalities, renal, autoimmune, and orthopedic issues are far less common. In terms of neurological conditions, again, we talked about this already quite a bit. A majority having microcephaly or a small head size. We have a varying degrees of sort of low muscle tone or high muscle tone, even some who've been diagnosed with cerebral palsy or coordination. So some general sort of movement and tone conditions that we do see across, but how exactly those are defined tends to be fairly variable. About half of the individuals in our sample do report some type of seizures. Again, I do want to remind all of you that some of these might not be mutually exclusive. So we do have some individuals who might be reporting more than one seizure type, but about half reporting grand mal seizures primarily. In terms of seizures, so we are trying to track data over time now about how do seizures evolve. So we're able to combine our seizure survey data. So we have 20 individuals who've completed this baseline seizure survey, and we are trying to continue to collect the seizure survey on a regular basis. We adopted and adapted the American Epilepsy Society seizure survey to complete this. So for those who did have seizure, the range is between 1 year and 5 years, with most individuals developing seizures at about 3. About half of individuals with those seizures have had to take medication, and all of them do continue to take some kind of medication. And all but one of them, about 10 who've ever had a seizure at all were able to achieve some kind of complete seizure control or complete remission, usually around about age 5. Heart conditions are not remarkably common, although we do have it reported in some individuals, including the ASD, stenosis. And we do have some surgeries. Some of these might include things like minor dental surgery, things like that. We do have most commonly a GI tube placement for our kids. Endocrine issues, largely we're seeing individuals who have short stature, and along with that, failure to thrive, so not gaining enough weight in infancy. Vision problems we see in at least about 1/4 to 1/3 of individuals with DYRK1A, often being nearsighted or having crossed eyes. I really appreciated what Sarah had said earlier about sort of the value of making sure that we're dealing with these things that help our children actually access therapies. So things like how well can your child see and how well can your child hear are really, really important to be able to access therapy and access school. So even though it's not the majority of individuals that are reporting some of these problems, sort of the thing at the top of my list to make sure that children are getting visits to their eye doctor or going to an audiologist to make sure that that's not a contributing factor or complicating factor to their developmental disability. We have a number of individuals who are using medications, most commonly for seizures or GI problems or for sleep. In terms of seizure medications, so we do, there's no really one medication that seems to work for everybody. We would like ultimately that, through some kind of some precision medicine that we would be able to move to certain medications quicker and say this is the one to try first. However, we don't really have all that information with DYRK1A right now, especially given that our relative sample of individuals who've reported seizures and are using medications is fairly small. Okay, so let's talk about the Vineland. I've mentioned this multiple times, so let's see exactly what we can see. So let me orient you first to this. So this is a little bit of we're trying to build this idea, like with the natural history studies, what does DYRK1A look like over time? So what you can see down here at the bottom is the child's age in years, so their chronological age. And this is sort of how we would think about their level of functioning in years. So we might have some kids, and all of these blue dots are individuals who have completed the Vineland. So you might take this individual who is 12 years old and look here at their individual dot and see that in terms of expressive language development, that we would estimate their functioning to be at about a 3-year level. This orange dotted line is the milestone that's associated with a child who's about between 1 and 2 years old, which is using single words to request. And then here, this dotted line is the milestone that's associated with a child who's about between 4 and 5 years old, which tells you about an event. And you can see here that this blue line is what we would expect in typical development, such that, a child who's at about 2 years old would be performing at a 2-year-old level. You can see in DYRK1A that children do typically make progress, although it tends to be much slower than what we see in kids who are developing more typically. What we do know at this point from the data that we have is that the majority of individuals with DYRK1A are using some basic language by about age 4 or older. There might be a subset of individuals who are not developing verbal language or are not using verbal language, but here's what we know, at least from the data that we have. So here's another little bit of a plug, although the Vineland is difficult to complete, but it can give us some information, again, about that natural history, what's the typical course, and it's also why we ask you to do it every year, so we can track your child's progress. Okay, so and here's the here is information about self-care. So here at age about 1.5, almost 2, we expect children to be feeding themselves. At about age 4, dressing themselves. Again, we continue to see progress. In none of these cases do we see children necessarily going backwards. We see progress. It's just more slow and steady. It's not at the same rate. It's not going backwards. In the majority of kids, we're not seeing a frank plateau. It's just more slow and steady. In terms of social skills, again, we know that about half of our children also are impacted by autism. So we do see a little bit of a slower slope here, and that individuals do tend to need more higher support for social interaction. And then looking at gross motor development. So this is an area of relative strength here for our kids with DYRK1A. So motor skills are delayed for many individuals. However, over age 4, for at least the individuals that we have data on, typically we do see that those children are walking. Looking briefly here, as I'm running out of time, I know, and I want to make sure I save time for emotional and behavioral concerns for children who are 5 years and younger. These are the types of concerns that you're most commonly reporting. So being hyperactive, having difficulty waiting, mouthing non-food items, and again, just having some general inattention. For children who are a little bit over, obsessions, I think you might recall that we did see one individual who was diagnosed with OCD being more impulsive and hyperactive, or ADHD type behaviors, and maybe being perseverative or repeating behaviors over and over. So overall, just to sum up, and I can take maybe a brief question or just go ahead and move on to our colleagues in the TIGER study. So our generally common issues are this intellectual disability or developmental delay with associated language impairment as well, about half also showing autism with seizures, microcephaly, and different tone issues and failure to thrive. And then other less common, but things that we have seen, low tone, other movement like cerebral palsy, some heart disease, although less common, some problems with vision, hyperactivity, and then other obsessive behaviors. Okay, I will go ahead and stop there. Do have Facebook and YouTube and Twitter to engage with us. We also have our DYRK1A Simons Searchlight group. If you're not a part of that, it is separate from the general family group if you'd like to get some updates from us.